TurboCHO expression system. In today's webinar, I will first talk about what is recombinant antibodies and how does it be generated, and then after that followed by how to select a suitable host expression system. After that, I will discuss why CHO expression system is still the favorite workhorse for recombinant antibody productions. Lastly, I will discuss what are the common challenges which scientists and researchers face today and what is the optimal high throughput solution from GenScript can help you to solve your problems in antibody productions. First, I will give an overview of the recombinant antibodies. Antibodies are proteins that belong to a family of proteins called immunoglobulin. Antibodies are produced naturally by lymphocytes, generally in response to infections by microorganisms such as bacteria or virus. The first research leading to the discovery of antibody was in 1890 by Emil and Shibashabura, transferred serum from the immunized animal against diphtheria to animals suffering from it and established the serum therapy. In year 1900, Paul proposed the site change theory, where he hypothesized that site change receptors on cells bind to a given pathogens. He was also the first to propose a model for an antibody molecules in which the antibody was branched and consists of multiple sites for binding to foreign antigens and for the activations of complement pathway. In year 1948, S3 actually demonstrated that plasma B cells are specifically involved in antibody productions. After that, in year 1957, Planck and David has developed the clonal selection theory. They stated that a lymphocyte makes a single specific antibody molecules that is determined before it encounters an antigens. But by 1959, Jarrett and Rodney independently published the molecule structures of the antibodies, for which they were later jointly awarded the Nobel Prize in 1972. In year 1973, the first atomic resolution structures of an antibody fragment was published, and this was quickly followed by the inventions of monoclonal antibody in 1975 by George and Kaiser, called hybridoma technology. This is the start of the modern era of the antibody research and discovery. In year 1984, Morrison and Neuberger independently developed the first chimeric antibody. The immunoglobulin gene from a mouse hybridoma cell line was cloned into a vector to be manipulated. Morrison and the team replaced the mouse FC fragments with a human XC fragment. Neuberger and the team expressed the plasmid in a myeloma cell line called J558L. In year 1990, antibody engineering technologies were established. After that, more and more therapeutic antibody has been approved and come out into the market. These are some of the therapeutic antibody examples come out from each year. Hybridoma technology is the primitive and most fundamental and successful methodology in the fields of monoclonal isolations. It relies on the B cells that are mature in the secondary lymphatic organ in response to an antigens. These antibody-producing B cells are then harvested from the mouse and fused with the immortal B cell called myeloma to form a hybrid cell line called hybridoma. It is actually cultured in the HAT media to select the positive cells and go on to the clonal selections and expansions for the monoclonal antibody generation. This technology is quite robust and useful in the discovery of thousands of antibodies for different applications. However, ethical, quality, and reproducibility issues are always the main concerns in this technology. To try to overcome this problem, a new generation of antibody has been developed, which is the recombinant antibodies. Recombinant antibodies are generated outside the immune system using a synthetic gene. DNA sequence of highly specific and versatile antibody will obtained from the phage, ribosome, yeast ribosome, and then cloned it into the vectors, and then introduced into the expression hosts such as bacterial, insect, yeast, or mammalian cell for the productions of the functional antibodies. Each of these technology platforms has their respective advantages, limitations, and applicability, which I will discuss further in the next slide. Recombinant antibody provide many important advantages compared to the traditional antibodies. Recombinant antibody do not require animal immunization in the production process, as recombinant antibody are derived from the DNA. Other than that, recombinant antibody is quicker to produce than the traditional antibody. Hybridoma technology require around months, whereas the recombinant antibodies technology just require weeks. And recombinant antibody has an excellent batch-to-batch reproducibility and lot-to-lot consistency because they are biologically defined and can be exactly replicated. They can also be genetically modified to obtain a greater affinity to a specific antigen. For example, they can be optimized by using a molecular biology methods such as site error prompt PCR or directed mutagenesis to increase their functions. Besides that, they can be subjected to isotype conversion, which means that a recombinant antibody fragment can be converted into several species, isotype, and subtype by changing the constant domains. And they can be also conjugated with other molecules such as biotin, enzyme, and fluorophore for research purposes, and conjugated with toxins and drugs for therapeutic use. They can be produced in a different forms such as the minimized antibody as CFV, FAB, VHH, and so on, which are not available with the traditional antibody technologies. Recombinant antibody maintain the high sensitivity, high specificity, and low immunogenicity. Recombinant antibody can come into different formats, including canonical IgG-like antibodies and the bispecific antibodies. Bispecific antibody is an artificial protein that can simultaneously bind to two different types of antigens or two different epitopes on the same antigens. There are many formats of the bispecific antibody, but the two main categories are the IgG-like, which have the Fc regions, and non-IgG-like, which doesn't contain the Fc regions. Antibody basic structure is a Y-shaped molecules consists of the two heavy and two light chain folded into a constant and variable domains. And the FAB domain, which consists of two variable and two constant domains. The variable domains of the contents of three hypervariable loops known as the complementarity determining regions and the framework regions. And the complementarity determining regions that allow an antibody to recognize an unlimited numbers of the antigens. The constant regions at the track of the antibody include the site involved in the interactions with other components of the immune system. Antibodies are divided into 5 major classes, which are the IgG, IgM, IgA, IgD, and IgE, based on their constant region and structures and immune functions. Bispecific antibody formats include the full antibody size, which contain the Fc regions, and antibody fragmented size without the Fc regions, such as the scFv, FAB, VHH, and so on, and the complex larger than the antibody formats, including the bivalence and multivalence formats. Bispecific antibody with the Fc regions are useful for carrying out Fc-mediated effector function, such as antibody-dependent cell-mediated cytotoxicity and complement-dependent cytotoxicity. They have the half-life to the normal IgG. Bispecific antibody without the Fc regions, which reduce the non-specific binding from the Fc interactions and rely solely on their antigen binding capacity for carrying out the therapeutic activity. Due to their smaller size, these fragments have better solid tumor penetration rate, but they are also rapidly cleared from the circulations, leading to a shorter in vivo half-life. Till today, there are 3 bispecific antibodies has been approved by FDA which for therapeutic use. In recent years, Bispecific antibody has evolved to be the most preferred and effective methods of treatments in several diseases. The global bispecific antibody market is poised to grow in coming years due to the large numbers of products approvals and increasing awareness among the populations regarding to the availabilities of the targeted therapies. Besides, the high prevalence of cancer and the unmet need for the targeted therapy also contribute to the growth of the markets. Bispecific antibody are different from the canonical antibody given the fact that they have dual functional which target multiple disease-modifying molecule with one drug. Therapy with single dual targeting drug rather than combinations should be also less complicated for patients. In this figure, bispecific antibody bind to two different types of antigen. One of its arm bind to the tumor antigens and another bind to the CD3 determinants on the surface of the T cells. These cross-linking tumors and effector cells increase the proximity while simultaneously trigger T cell activations. The result is a targeted and highly effective tumor cell killing. The goal of this effect is to simultaneously target various targets involved in disease pathway and thereby increase the therapeutic efficacy. From a technological and regulatory perspective, this makes the developments less complex because of manufacturing, preclinical, and clinical testing is reduced to a single bispecific antibodies. Next, I would like to talk about how to select a suitable host expression system for recombinant antibody expressions. These are the widely used host expression platform for recombinant protein in the market. They are bacterial, yeast, insects, and mammalian system. In the eukaryotic system, such as yeast, insects, and mammalian cell, they have the post-translational modifications, but not in the prokaryotic system, such as bacteria. Therefore, select an appropriate expression system is a crucial factor to produce correctly folded proteins. For antibody manufacturing process development, maintaining desired quality attributes while reducing time to the markets, maintaining cost effectiveness, and providing manufacturing flexibility are the key issues in today's competitive markets. These are the three main expression systems used in the industrial, which are the E. coli, insects, and mammalian cell. And E. coli does not have the post-translational modifications. Both of the insects and the mammalian cell have the post-translational modification, but insect cell have a simple post-translational modification. Therefore, post-translational modifications are important factors to be considered when choosing an expression system. It actually affects the protein structures and function. In terms of the cell growth rate, E. coli is growing faster compared to the insects and mammalian cells. Mammalian cell will be medium to fast depending on what kind of proteins you are synthesized. And based on the complexity of the growth media, E. coli is less complex compared to the insects and mammalian cell, which need to be optimized various conditions. Therefore, in terms of the pricing wise, E. coli will be cheaper compared to the rest. Other than that, expression level of E. coli is higher and mammalian cell will be moderate to high, and insect cell will be typically in low amount of expression. The recombinant proteins were synthesized and secreted to the periplasm of the E. coli. However, insects and mammalian cells are able to secrete to the media, which is easier to harvest the protein and perform the purification step. Talk about the endotoxin level. It is important for the therapeutic drug development. E. coli is a gram-negative cell, therefore endotoxins is always the concern when using this system, but not in the insects and mammalian cell. E. coli is suitable to express the antigens, intracellular proteins, and two enzymes, while in insects, cell is suitable to express the intracellular and secretory proteins. Complicated target proteins such as kinase, protease, and transmembrane proteins. Mammalian cell is typically for secretory proteins and the antibodies. Our GenScript provides a series of high-throughput recombinant protein expression platforms, such as bacterial, insects, mammalian, and yeast. We have more than 18 years of synthesis experience. Our expert scientists and dedicated account managers are invested in helping to maximize the success of your research project and will guide you every step of the way. We successfully deliver 100,000 recombinant proteins projects and able to have a 98% of expression success rate. Besides that, our another new production facility in Singapore is set out in this year February, which will help us to serve you better. Choosing a suitable expression host not only will help you to improve the expression yield, but also will affect the protein functionality. There is studies show how does different expression system impacts the bispecific antibody not into host assembly. In bispecific antibody assembly, half antibody can be expressed in the E. coli or CHO host cell, and follow cell culture expressions are captured from harvested cell culture supernatant with similar techniques as those used to capture the monoclonal antibodies. The decision of which host cell type to use is driven largely by manufacturing capacity. Productions of biopharmaceutical in CHO is well understood process with a long history of use in the industry. Although the numbers of products quality attributes are affected by the choice of the host cells, major impacts was seen in the bispecific antibody assembly is the effects of the heavy chain CH2 glycosylation. Protein glycosylation is a post-translational modification that is absent in the prokaryotic cells such as E. coli, but present in the antibody produced from the CHO cell. And antibody glycosylation is known to promote the acid effector's functions. Glycosylation improves the antibody stability by raising the melting temperatures of the CH2, and interestingly also increase the assembly of bispecific antibodies only when both half antibodies are glycosylated. Therefore, host expression selections is crucial for bispecific antibody assembly. FDA-approved therapeutic antibody lenalidomide is a bispecific CD19 and CD3 T-cell engager construct using bispecific T-cell engager antibody technology, and CD19 is expressed nearly ubiquitously in the B-cell lineage beside than plasma cell, and therefore this drug is an option for most patients with relapsed or refractory B-cell acute lymphoblastic leukemia. Upon binding onto the both CD19 on the B cell and CD3 on the T cell, this drug promote the activations and expansion of the CD8 cytotoxicity and CD4 helper T cell, resulting in the lysis of the malignant and normal B cell. There is a comparison study of different host expression systems on these bispecific antibodies. Bispecific antibodies was expressed in the E. coli and on different CHO cell, and bispecific antibody produced from the CHO cells had higher affinity and specificity for both CD3 and CD9 antigen than the one expressed in the E. coli. Therefore, this also proves that choosing a suitable expression system to express the target protein with all post-translational modifications have great importance. Mammalian cells process a conserve secretions and folding machinery. The resulting antibodies are expecting to gain the specific post-transcriptional modifications that enable the immune activations and target recognition mechanism in the mammalian system. Now, I would like to discuss why CHO expression system is still the favorable workhorse for the recombinant antibody productions. It has been more than 3 decades since the first monoclonal antibody, muromornate CD3, was approved by US FDA in year 1986. The increasing importance of therapeutic antibody is apparent as therapeutic antibodies has become the predominant treatment modality for various diseases over the past years. Due to the recombinant antibody technology advance has made the discovery and development of the monoclonal therapies quicker and more efficient, the global therapeutic monoclonal antibody market is expected to generate revenues of 300 billion by 2025. And last year, May, 100 therapeutic antibody has been approved by FDA, and majority of them are in the cancer therapy. From year 2014 and 2018, 84% of monoclonal antibody therapeutics was approved and produced by using CHO cells. Several favorable properties have driven CHO cells increased use in the bioprocessing, including their resilient to growth conditions, resistant to the virus infections, and the high protein synthesis capability. So what is CHO cell? CHO cell is the Chinese hamster ovary cell that derived from the epithelial cell of the ovaries of Chinese hamster. They are widely used for biological and medical research and commercially used in the productions of the recombinant therapeutic proteins. This is a significant amount of work that has been done to generate engineer productions whole cell to improve or modify the product quality or improve the cell robustness. The first CHO cell line was established in 1956. CHO ori cell actively divide and do not exhibit the limitations on doubling times observed in the primary cell. After that, in 1968, CHO ori was cloned to CHO K1. This cell line is missing a chromosome which carry a gene necessary for glycine biosynthesis, paving the way for selection methods. In the 1971, CHO-S was generated and it adapted for the growth in the suspension liquid culture. This cell line is ideal for scale-up and growth in the large-scale bioreactor. In the 1980s, CHO-DXP11 is CHO cell lacking a DHFR activity in one locus and missense mutation in the others. DHFR deficient strains cannot grow unless the transfectors with functional copies of DHFR or intermediate supplements with the thymidine. Thus, transfecting DXP11 cells with a functional DHFR gene attached to a genes of interest enables selections of the genes only carrying their genes of interest by growing them in the thymidine-free media. In 1981, CHO-GS is using the selections of recombinant cell line using a stepwise increasing in the MTX, which is the antagonist of the DHFR concentration in the culture media, resulting in the amplified copies of the transfected DHFR gene together with the genes of interest. Such induced gene amplification usually increase the productivity of the genes of interest. In 1983, CHO-DG44 was generated, which is the containing full deletions of the two DHFR loci, and CHO-DG44 eliminate the problems of the CHO-DXP11, which could spontaneously revert back the functional DHFR enzyme. This new CHO cell line makes selections for the gene of interest always possible. Due to this, CHO-DG44 are amongst the most widely used CHO cell for the industrial protein productions. In year 1986, human tissue plasminogen activators, marketed as the Activas, is approved by FDA, become the first therapeutic proteins from recombinant mammalian cell to obtain the market approval. In year 1989, CHO K1SV was developed and it is a suspension protein-free adapt CHO K1 derived from the utilizing the glutamine synthase gene expression system. CHO K1SV express the glutamine synthase enzyme endogenously. Therefore, positive transfectance was obtained under the dual selections of methionine sulfosamate and the glutamine-free media. Until today, more than 100 new recombinant protein therapeutics have been approved by US FDA or EMA. With the increased interest in the virus diagnostic and the treatments, CHO cells are again demonstrating their versatility. While CHO cell is predominant in the mammalian expression system, HEC293 is actually derived from the human cell line, therefore is more accessible for the human's virus compared to the CHO cell from the hamster. In terms of the biosafety, it is also very crucial in the pharmaceutical drug development. Compared to transfection efficacy between the HEC and between them, HEC is easy to be transfected, but sometimes CHO is not. Other than that, CHO cell have a faster growth rate compared to the HEC293 cell, and CHO cell are able to grow in the serum-free media compared to HEC293, which is not able to. And as mentioned earlier, post-translational modification is important in protein folding and function, which will impact the protein functions afterwards. Since HEC293 is derived from the human cell, therefore it can fully replicate its human-like post-translational modification. But CHO cell can adhere many of the post-translational modification, but sometimes might have slightly different. Due to a large degree of familiarity, and CHO cell have been the predominant choice for the productions of many recombinant proteins. These choices are augmented by several glycoengineering strategy to humanize the CHO cell. After the host has been decided, we have to think whether we need to perform a transient or a stable transfection depends on the time frame and the ultimate goal of the experiment we wish to conduct. Both of these transfection methods involve getting the foreign target gene into the cell. In the transient transfection cell, the foreign DNA does not integrate into the host genome. It does not replicate and is eventually lost throughout the cycles of the cell division over several days. Transient transfection is good if you need it fast, and typically 5 to 7 days from the point of DNA transfections. And stable transfection also begins with transiently, but through a process of carefully selections and amplification, stable clones are generated. In the stably transfected cell, the foreign gene becomes part of the host genome and is therefore replicated. Descendants of these transfected cells also express the foreign gene and resulting in a stably transfected cell line. Because the stable transfection of cell is longer and more arduous process, therefore the duration is longer, which is more than 3 months. During the initial short-term selection process, when research quantities of the recombinant antibodies are needed, rapid methods of recombinant antibody productions are required, but it might have a slightly various in terms of the batch-to-batch productions. For long-term study and productions of the therapeutic antibody, stable gene expression is the preference option as it allows for greater process consistency and control of the final product's quality. Lastly, I would like to discuss what are the common challenges scientists and researchers facing in the antibody productions. So what are the common challenges scientists and researchers facing in the antibody productions nowadays? For example, how can I get a better quality antibody products? I am encounter a budget issue, and what is the yield of my antibody is always so low, and I don't have enough capability to synthesize so much of targets, and my project or thesis deadline is coming. I don't have a sufficient time to finish the productions. Don't worry, the optimal solution for all these challenges can be solved by our new next generation TurboCHO expression system. Our production team is able to produce 10,000 targets per month. It can be produce high yield and high purities of your antibody from milligram to kilogram. It has been faster turnaround time, which just need as short as 2 weeks, including gene synthesis for typical antibody productions. And we have a full range of QC and characterization tests available to further identify the quality of your products. And it can be produced in our Singapore and Nanjing facility. It is more cost effective which enable small scale to a kilogram level protein production based on the same expression system for the best consistency and minimal surprise during the downstream step. We also utilize the unique QR code to track the production to ensure there is no error rate occur. This system is able to scale out and finish by CHO, which will save time to optimize the condition if you want to move on for the large-scale productions. Our next generation proprietary CHO expression system is suitable for all types of proteins, antibody species, and isotype, and the antibody fragments as well. You can check out more in our GenScript webpage under new TurboCHO mammalian recombinant proteins and antibody expression service. If you have any queries, please feel free to contact us directly. Our GenScript provides us one-stop solution for recombinant protein productions. What customer need to do is to just provide us the sequence of the proteins or antibody sequence, and our production team will evaluate and perform the codon optimization to improve the gene expressions. After the gene synthesis and construct generated, expression construct will be transfected to the expression host and scale out correspondingly volume, and proceed with the protein purifications and analysis, and then the final products will be sent to you in a very short turnaround time. Here, I would like to share some of the case study which have been done in this our new next generation TurboCHO Express platform to produce the recombinant antibodies. These are the SDS page result with the antibody samples of rabbit IgG, human IgG1, and human IgG4 run in the reducing and non-reducing condition. We are able to produce a purity around 99% or more in these recombinant IgG. Here is another example of the bispecific antibody using nodes into hosts and the cross-match techniques, which expressed in our new next-generation TurboCHO Express platform. This is the SDS page result run in the reducing and non-reducing condition. We are also able to produce more than 99% purity in these bispecific antibody productions. Customer just need to provide us the antibody design sequence and the ratio of the plasmid for this bispecific antibody. Then we can help you to start from gene synthesis to the antibody productions. In our next new generation TurboCHO expressions platform, not only can produce proteins, canonical IgG or bispecific antibody containing the Fc region, we also can express and produce the antibody fragment, for example, scFv or VHH. The products of these antibody fragments were run in this SDS page gel with reducing and non-reducing condition. This scFv is tagged with his tag and purity is around 99% after one step of his trap purification. For the VHH is tagged with SC tag, and we are able to produce it and purity more than 99% after the protein air purifications. That's all for my today presentation. If you have any inquiry, please feel free to contact me or the COMA, and thank you for your attention.